change this or to replicate this plasmid. So we need the so-called helper phage. The helper phage, it contains all the information for the synthesis of new bacteriophages. So the 3 types of genes, you remember, the morphogenetic genes for the assembly of the bacteriophage, the structural genes that are responsible to make the structure of the phage, and then you have the blue ones, which are replication genes that will be expressed, and these replication genes will then go and recognize the packaging signal and the origin of replication for the replication of the phage mate. But to make sure that these 2 genetic material are not packaged within the bacteriophage, we disable the origin of replication of this helper phage. That means this genetic material, the genome of the helper phage, won't be packaged because it doesn't have the packaging signal. But when replication starts, when we make the DNA replication genes, they will recognize the packaging signal and the origin of replication of the phage mate, and then the phage mate is the one that is going to be packaged onto the new bacteriophage. And this is shown in this diagram here. So you allow infection of a helper phage, and then you transform the bacteria with the phage mate. The phage mate will be changed to single-stranded DNA by the replication genes. You remember the G2 protein and the G5 protein that comes from the helper phage, and then the G2 and the G5 will recognize the single-stranded DNA, and then it will be packaged along with the structural proteins that are being made from the helper phage. So we have made a new phage that contains only information or the genetic material of the phage mate instead of the helper phage. So what you can do next is to genetically engineer the phage mate to contain, for example, the gene number 3 protein, open reading frame, or the gene number 8 protein that has been, in which DNA has been introduced, and this DNA will be expressed along with these 2 proteins within that protein to make the so-called fused protein. So you'll have the structural proteins, but also at the end of that protein, you'll have a series of peptides, and these peptides will be fused to the structural proteins, and there will be the peptides of interest that we are expecting to harvest. So in order to harvest, for example, if we have been able to introduce the genetic information within gene number 3 or gene number 8, and a peptide is displayed along with these proteins, in order to select for this foreign peptide that we have been expressed, assume, for example, they are being recognized by a certain monoclonal antibody, we use a process called biopanning to harvest for those bacteriophages that only contain the peptide of interest. Imagine you had a huge amount of random library of DNA, genetic material, that you have been introducing to the phage mate, and you have fused it with the open reading frame of the G3 or the G8. You won't be able to know which bacteriophage contains the peptides of interest. In order to know the bacteriophage that contains or which express the peptide of interest, you have to do biopanning. So you have a column that is in which a monoclonal antibody that specifically interacts or recognizes that peptide is immobilized into the column, and then allow all the different types of phages to pass, and then only those phages that contain that peptide of interest will be trapped with the monoclonal antibody. And once you elute the other phages, they will go through, and then you start eluting only the phages of interest, and you amplify that by infecting bacteria. So you produce other colonies that contain huge amount of bacteriophages, and you can repeat the process of binding to the column. In this way, you enrich your library, or you enrich the colon or the type, only the type of bacteriophage that express the desired peptide. Instead of biopanning, you could use other tests, for example, an ELISA test, like shown in this slide here. The ELISA test, in the ELISA test, you will immobilize a monoclonal antibody on the surface of the plate, and then you will allow different types of colonies to go into each different well, and then only those bacteriophages that contains the peptide of interest will be able to interact with the monoclonal antibody, and then you would add in other antibodies that recognize other structural proteins on the surface of the phage, and then these antibodies will be conjugated with, for example, an enzyme, horseradish peroxidase, and when you add a substrate, that substrate will be cleaved by the enzyme to produce a product that gives color, and so you will only go for the wells that have produced that chromogenic substance, and this will be the peptides of interest. So there are some rules that I want to tell you that are important during phage display. How do you introduce the DNA onto the open reading frame of these structural proteins? If you would introduce DNA at the middle of the open reading frame, you disturb the open reading frame, so such structural proteins cannot be produced. So there are areas that are advised to do the introduction of the genetic material while not disturbing the open reading frame. So there is desirable to introduce your foreign DNA between the signal sequence. The signal sequence is the amino acids that will target the synthesized or the translated proteins onto the cell membrane of the bacteria. And so you introduce the DNA just between the signal sequence and the open reading frame of the caught protein gene, or at the end of the caught protein gene. This is very important because in that way you don't disturb the production of the caught protein. And in order not to disturb the open reading frame, you have to introduce DNA in multiples of 3. Why should you introduce DNA in the multiples of 3? It's because each codon is made up of 3 amino acids. So if you would introduce, say, 4 amino acids, then the codons downstream the introduction are going to change, and so you will no longer make such a structural proteins. So there are vectors that are meant for phage display. For example, here I have the FD-TET DOG1, and it contains different molecules that or genetic information that's important for cloning and expression of peptides. There is another vector here, the PHEN1. As you see here, there is the so-called histidine tag, and the histidine tag will be important in purifying such proteins. And then it contains the MyC tag that is also important in selecting or trapping the bacteriophages that are expressing the protein. And then you have the gene number 3, structural protein. So I hope that I have introduced to you one of these this molecular biology tool that's very important in genetic engineering and molecular biology to express foreign peptides on bacteriophages, and you know why we use bacteriophages. I have said, maybe I would summarize again, bacteriophages are used because they contain a self-replicating machinery, and only one bacteriophage will infect a certain bacteria, and so we expect that if we have introduced certain nucleotides, they will code for information or a peptide that is of interest, and only one bacteria can be infected by a bacteriophage, so we are able to get such a good colony of a good population of bacteriophages from one bacteriophage. So there are different applications for the peptides that we desire. For example, you could use them in the diagnosis of different kind of infections. So you make peptides that are recognized by certain antibodies. Or you can use to study protein-protein interaction. For example, if you do not know which protein interacts with which protein and at which region, you'd want to put a certain peptide on a bacteriophage structural protein, and then do the bait and prey trapping such that you can understand which protein interacts with which protein in molecular biology. I thank you for your attention, and I welcome you to continue listening this series of molecular biology and genetic engineering lectures as we build you to be future professionals in diagnosis of infectious diseases and genetic engineering for improving animal and human health. Thank you very much for your attention.